I'm standing amongst the beautiful perennial collection at the New York Botanical Garden, surrounded by lush leaves and strong branches. This is collectively called the vegetative shoot system, which is describing the above ground, non-reproductive portion of a plant. I'm Dr. Joyce Onyenedum, and I'm a botany professor at NYU. Welcome to Build a Plant, where we discuss the hot topic of plant anatomy. The vegetative shoot system is comprised of stems for structural support and leaves for photosynthesis. Without shoots, plants are not plants. So how do they form? Interestingly, individual plant cells and the entire shoot system are similar in how they grow. First, they elongate and then they thicken. In a shoot, the elongation phase is called primary growth because it occurs first. This is the topic of today's video. We will cover secondary growth, the thickening phase, in the next video. Indeterminate primary growth happens continuously throughout a plant's life, made possible by the activity of a tiny pool of stem cells at the tip of shoots. This powerful pool is called the shoot apical meristem, aka the SAM. In seed plants, the SAM is externally dome-shaped, like in this Coleus plant, but internally, it's like a factory, dissected into different departments with distinct functions. They work together to initiate all cells, tissues, and organs in the vegetative parts of plants during primary growth. The SAM dome can be dissected into layers or zones, depending on who you talk to. We'll begin with the tunica-corpus scheme, which separates the SAM into distinct layers based on the plane of cell division. Here, the tunica refers to the outermost tissue layers that exclusively divide anticlinally, that is perpendicular to the meristem surface. The tunica layers typically produce the epidermis. Just below these layers is the corpus, a body of cells several layers deep, where cells freely divide in various directions to add to the bulk of the shoot axis. The cytohistological zonation concept adds a bit more complexity, separating the SAM into zones based on location, rates of cell division, and the exact tissues and organs that they produce. At the very top is the central zone, where two equally important events occur. First, new cells are made to replenish themselves as the initiation site. Second, as more cells are generated at the initiation site, older cells are spit out of the central zone to feed into the two neighboring zones. Those zones are the peripheral zones on the flanking regions, which gives rise to the cortex, procambium, and leaf primordia, as well as the centrally located rib zone, which produces the pith. Classical anatomy also has terminology for the immediate derivatives of the SAM, tissues that have begun to differentiate but haven't quite finished cell division yet. They are called the primary meristematic tissues, or primary meristems. The protoderm is the primary meristem that gives rise to the epidermis, the external tissue layer of the plant. The procambium gives rise to the primary xylem and primary phloem, the vascular tissue that transports water and sugar. The ground meristem generates both the pith and cortex, which are supporting cells that serve numerous metabolic roles such as starch storage. Together, they're known as the ground tissues. Although researchers may be using different names, many of these concepts do in fact overlap. For instance, the protoderm is derived from the outer layer of the tunica, and the rib zone only overlaps the corpus, while the peripheral and central zones span both tunica and corpus. Critical to the establishment of primary growth is the formation of vascular tissue, the primary xylem and primary phloem. The general arrangement of the primary vascular tissue in the shoot is known as the stellar anatomy, and it's organized by the activity of the procambium at the shoot apical meristem. The eustele is the most common stellar anatomy, present in some form across the shoots of all seed plants. This is why it's hammered so much in your introductory botany courses. In a eustele, the procambium generates individual units called vascular bundles that encircle the stem, sandwiched between the pith internally and the cortex externally. Everyone who has taken botany has seen a eustele. It is also the stellar anatomy in the plant model species Arabidopsis. So today, we will highlight how developmental modifications in plant evolution have led to novel stellar anatomies with significant functional consequences. Within the large angiosperm orders, Caryophyllales, we find numerous species with medullary bundles. Those are additional vascular bundles arising in the pith from procambium left behind by leaf, bud, or thorn traces. The number of medullary bundles can be plentiful, like the columnar cacti, where they're hypothesized to be associated with the evolution of having a large pith in such big succulent plants. Recent studies have even shown that medullary bundles are responsible for roughly 50% of fluid conductance in the herbaceous species Allionia incarnata. You may notice that these medullary bundles are eerily similar to the stems of monocots, which have scattered vascular bundles in an arrangement known as an atactostele. However, the vascular bundles in monocots arise from a different developmental process and have distinct structure. Let's discuss another odd yet important evolutionary change to the classical eustele. Vining plants in the family Sapindaceae have a rope-like stem. How does it form? The young shoots that are experiencing primary growth have a eustele, but instead of the vascular bundles forming a ring towards the center, the bundles are way out to the edges of the lobed stem. By dislocating those bundles to the periphery, the vascular system is dissected, and the vascular tissues become compartmentalized. These separate units lead to a rope-like look in mature plants. Let's recap. The vegetative plant system is initiated and sustained through a process called primary growth, which involves elongation of the main shoot axis and the generation of new tissues and organs. Primary growth is executed through a tiny yet powerful pool of cells sitting at the dome of shoot apices, called the shoot apical meristem, or the SAM. The anatomical literature and molecular literature may be using different terms to describe the SAM, but they describe spatially overlapping regions that produce the epidermis, primary vascular tissues, and the ground tissues. The eustele is present in all seed plants, yet we find odd anatomical deviations from the norm, including medullary bundles and the rope-like stems of vines. And finally, while primary growth describes how shoots elongate, in the next video we will cover how plants bulk up through the developmental process called secondary growth.